Okay. Um, why don't we go ahead and get started? Okay. So I am Sue Biggins, and I'm in Basic Sciences, and it's a huge pleasure for me to introduce not only one of my colleagues today, but two. And there are two really talented PIs in the computational biology program. And the reason you're hearing from both of them is because one of the goals of this Science for Staff talk is to illustrate the strength of collaboration. So modern science is more complex than ever, and it's become more and more important to collaborate to make discoveries. Collaborations allow investigators with really different expertise to apply it to a similar problem and propel science faster. And one of the really great things about working at the Hutch is that the investigators are really encouraged to collaborate. And Jesse and Trevor are a great example of this and are going to tell you about that today. Okay, so before they talk about their work and collaboration, I just want to tell you a little bit more about them. So they're both associate members, as I said, in the Comp Bio program. Trevor did his PhD at Harvard in the Hartle Lab, and then he did a couple of short postdocs in epidemiology, and then there was a virology one as well to get experience understanding infectious disease and viruses. He joined the Hutch with an appointment in VID in 2013, and he's really done fantastic work since then working on viruses. His interests are in figuring out how virus populations spread and evolve to ensure that they survive. And one of the really cool things he's pioneered is a website called Nextstrain.org. And it's actually real-time virus tracking in the world. So you can actually see where mumps is right now and what country has the most and which type, or Ebola. And so it's actually very cool. You should check it out. He's been recognized already with some really prestigious awards, and he won the Open Science Prize for the website. And he was also named a Pew Scholar, which is one of the most competitive and prestigious awards that an early stage scientist can get. Jesse is similarly amazing. He started here in 2011, and his primary appointment is in basic sciences. And he's also interested in viruses and how they evolve. He did his grad work with David Baltimore, and then he was a postdoc with Frances Arnold, and she was one of the Nobel Prize winners this year. And that gives Jesse the unusual distinction of having done all his training with Nobel Prize winners. Like Trevor, he's been really successful and made a lot of discoveries that explain why we can't make an effective vaccine against the flu, and you need a flu shot every year. He's also a notoriously great mentor, and currently, I think he actually has more grad students in his lab than most faculty have ever had in their career. And like Trevor, he's been recognized with many national awards, and this year he was named an HHMI investigator. So we are very fortunate to have both of them here at the Hutch, and I know there's going to be a lot of great science coming from these labs in the future, and so we'd love to hear about it now. I guess Trevor is starting. Okay.
Okay, can you all hear me okay? Yes. So thank you for the very kind introduction, Sue. Jess and I do have a number of collaborative projects, and he's been a really great collaborator and mentor in the last 5 years that I've been here. For this talk, however, it's not one of the collaborative projects. I thought it better to mix things up and not just talk about flu for the full span of time. So in kind of thinking about this and thinking about kind of using evolution, what I always come back to is phylogenies or phylogenetics, which is basically just showing you how one thing is related to another thing. And I love this example here where we can make a phylogeny or an evolutionary tree of the English monarchs. This is the Tudor lineage, and you can see how different folks are related to one another. We can zoom out a bit, and so this is kind of the tip of this tree here. This is by Haeckel in 1879, kind of showing all the different animal relationships. We can zoom out further and look at kind of what we've learned from doing molecular sequencing and look at the entire tree of life and then see that kind of here we are as humans at this very tip of all of the tree of life of kind of this everything being linked to everything. And what's really interesting, though, is kind of generally, if you look at the literature, these phylogenies are just kind of telling us about history, and we know what happened. However, even from the very beginning, we have this figure from Darwin. This is the only figure in The Origin of Species from 1859. And it's showing his idea of a phylogeny, even before, before it was possible to actually produce one, where we have a species here. So this is time going up the page. And here we have kind of a progenitor ancestral species back here. And then kind of different forms emerge along this axis. And then what happens here is that the species that are more different from one another, that kind of use different niches and different resources, are the ones that survive and propagate, and the ones that are kind of in the middle here are forced out. And so you get this kind of branching process as ecology unfolds. And so Darwin had this like really keen insight before we kind of had any actual data to work with here. And so what I work a lot with is trying to take these phylogenies that we can look at from data and then learn something about this underlying process. And I should also say that I'm happy to, if you have a question, just interrupt me, and I can take a few at the end of my section. So as an example of what we can learn about process here, so this is a little, see if this works. Yes, make it bigger. So here's a little toy example where we have, say, two populations. There's like fish in one lake, in the orange lake, and fish in the blue lake. And you can look at how, or maybe upriver, downriver, and you can sequence things and look at how they're related to one another. And that's telling you something about this kind of branching relationships. And what you're kind of seeing here is each time an individual replicates, we get kind of a split. And there's a small chance of orange individuals moving to the blue area, or blue individuals moving to the orange area. But I've set it up so that there's more likely to go orange to blue. And you can then generally see, I'm going to try to pause it, that as you kind of go up the page, we see that we go from kind of blue to orange rather than orange to blue. And we can then use the shape of the tree to kind of learn about how things are moving about because of this. So to show how that works, do I go back? Oops, oh, that was bad. So if we look at kind of that demo then, if you actually kind of play things out, it looks something like this, where we then have kind of yellow and blue here. And you can see that if we take individual, so this is kind of an individual virus or individual fish or any kind of biological entity here, and we trace it backwards, we see that even if we sample individuals in kind of the blue island or the blue lake, as we go back, they end up in kind of the yellow area. And so we can learn about this kind of patterns of movement and migration by looking at the phylogeny and learn about this kind of ecological effects. So this is kind of the very broad scale. What I'm interested in more specifically is using this kind of technique to look at how viruses and pathogens move about the world, because this is kind of something that's very relevant to us. And so if we look at swine flu in 2009, so swine flu emerged after there was a pig virus that was circulating in pigs in the Yucatan, and then that infected a person, and then that virus is able to spread from person to person. That first virus that infected that person from pig to human transmission event happened around March 2009 or February 2009, and then very quickly, within just 4 months, it had basically made it throughout the entire world. I'm going to play this again, and you can see kind of that early appearance and then very quick spread, eventually getting to even to remote parts of Asia just 4 or 6 months later. And so we want to be able to understand how that process happens and how viruses move about the world, but it's actually very hard to kind of look at that directly. There's no kind of, you have to kind of find these rare cases and have these giant surveillance systems. And what you can do instead is you can kind of look at the virus and ask the virus based upon its genome how it's spreading. And so kind of how this works as a toy example, so if we sequence a virus then, and we sequence several viruses, so these are different flu viruses that I have here. This is a flu virus from Hong Kong in 2017, virus from China in 2017, and so forth. And then what we can see is that there are going to be each of these different sequences of letters is one virus genome, is one virus sequence. And what we're looking for then is shared mutations. So this virus has this C mutation, T to C mutation. This one does as well. This one does as well. This one does as well. And so we can use those mutations then to know that those viruses kind of are more closely related than other viruses. And we can use these patterns of shared mutations to learn about what is related to what. So kind of how that works is, let's say we have an epidemic process. So we have one person infecting another person. And so this is a person that's sick with say, flu. And then over the course of 5 days, they're going to get a couple people sick here. So this person spreads it to this person spreads it to this person. After 5 days, that person has recovered, and they're no longer sick, but then this person spreads it to 2 more people, and so on. And this is generally how you get kind of these big kind of epidemics going on. But what we can do now is we can take some of these individuals, these infections, and then sequence not the human, but we can kind of take a swab from their nose and sequence the virus that they're being infected with. And then we can look for these shared mutations. So this purple star here is kind of the example that I showed before of the T to C, where this is a particular mutation that is shared by these 2 individuals, and that this orange mutation is shared by these 4 individuals. And so you can use these patterns of shared mutations then to construct a phylogeny. So this is the phylogeny for a toy example. What it looks more like in practice generally is something like this. This is a part of a tree from Zika virus, which I'll be talking more about in the rest of the talk. This was actually samples that Ali from the lab and Louise from the lab collected in Colombia. They went down as part of a collaboration with the Colombian NIH. And so we can see here that there are these viruses from Colombia, Panama, Dominican Republic, Colombia. And we can use that, like that example that I was showing you with the kind of orange and blue colors and the tree where it kind of goes from one region to another. We can use this to learn about how the viruses move the actual viruses move from one region to another. So we think then that there was a virus that existed in Colombia in mid-2015, and then it spread from there to Panama in one event, and it spread from there to the Dominican Republic in a different event. And so we can then use this kind of shape of the tree and where viruses were collected from to learn about how viruses are moving about the world. So generally, I think this kind of phylogenetic tracking has the capacity to revolutionize epidemiology, but much of that has just been too slow to really have a real impact because it takes sequencing things is hard, and it's always kind of delayed compared to actually how fast the outbreaks are happening. And so we're working to change that. So I want to talk about 2 different things. One is just the story of Zika's spread in the Americas as we've learned it from genomics, and the other is some of our work, as Sue mentioned, on Nextstrain and rapid phylogenetics. So for Zika is a virus that is almost always mosquito to human transmission. So someone that's sick with Zika will be sick for 2 or 3 weeks, generally 2 weeks, and then during that time, if they're bit by a mosquito, and the virus is going to be in their blood, the mosquito can draw some virus, and then when it bites a second person, transmit that virus between them. And so Zika has basically became a kind of world health emergency when it's discovered that if pregnant women were infected by Zika virus, they have a significant chance of birth defects and microcephaly. So the Zika kind of appeared, and it had been circulating at very low levels in Southeast Asia. We weren't really noticing it. But then there were some really large outbreaks in the South Pacific and French Polynesia around 2014 that were kind of isolated outbreaks and unexpected. And then there was this really huge outbreak in the Americas in 2015 and 2016, primarily in South America and Central America, just mainly travel-associated cases in the United States. And then since 2016, it's really died down because people have now kind of developed immunity to the virus. However, during this time, it was really unknown how the virus got to Brazil, how the virus got to South America, how many times did it actually migrate, and all of these kind of questions about basic epidemiology and the basic understanding of the outbreak were very unclear. So we did a project then to kind of investigate this. This was a big collaborative project with colleagues from the UK and also from Brazil. And Ali in the lab was also involved. And basically it took the form of a bus trip. So we took this bus, like a literal bus, and then drove it across the northeast coast of Brazil and stopped at these government diagnostic labs. So each kind of state in Brazil has a LACEN, which is their state-level diagnostic lab. And basically these diagnostic labs are receiving many blood samples from primarily pregnant women who were sick with Zika and they needed to, or sick with something like Zika, and the diagnostics lab role is to say whether or not it's an actual Zika infection. but basically the diagnostic lab wasn't able to keep up with things because they had so many samples coming in. So we were able to kind of just bring people and equipment and then like look through these samples and find and identify Zika positive samples. So then the diagnostic lab could report back to the clinician for the results of the diagnostic test. But then we also took the positive samples, took a portion of the positive samples with us for later sequencing. And so that sequencing that happened at the end of the study in Salvador and Sao Paulo with the Brazilian collaborators. And so just a couple fun photos. Here's the bus that we had. We have this wonderful little bus inception thing going on here. At the back of this, here, there's a kind of very small lab area that we're using to try to do some sequencing to kind of keep things separated between the diagnostic work that we're doing within the government labs and then the sequencing work that we're doing on the bus. Here's me doing my best to help out. Here, this is one of the more exciting developments that have happened recently. So this little thing that you're seeing here is now all that it needs to sequence anything, basically. I didn't bring it with me, I should have. It's this little kind of USB-powered device that's smaller than a phone that you plug into your computer, and then it has a little tiny pore on it that when you put in DNA, that DNA kind of flows through that pore, an electrical signal is generated that then you can read out that electrical signal as ATGC. And this is kind of remarkable technology. It's really enabling for what we're doing because the facilities in Brazil do not have the kind of great facilities that the Hutch has in terms of the sequencing capacity. And there are huge issues with expatriating samples. So Brazil doesn't want their Zika samples to leave for kind of good reasons. And so the kind of solution to this problem has become to bring the sequencing to the samples rather than the samples to the sequencer. And so this still requires a full lab infrastructure, basically, to make the thing that goes into the pipette to pipette into the MinION device, but it's something that was kind of not at all possible before this. This is actually doing sequencing in the hotel when the power had gone out, so it's by iPhone light. Okay, so that's the trip. What we saw just from these kind of diagnostic tests is basically that there was already something kind of funny going on in northeastern Brazil, where northeastern Brazil has a peak of Zika earlier on than the rest of the country. Whereas, so if you look elsewhere in Brazil or elsewhere in South America, it doesn't have that peak in mid-2015. So it looks just from the number of cases like something is going on in northeastern Brazil, different from elsewhere. And then really the sequencing results show this very clearly. So this is another phylogeny like the ones that we were looking at before, where we see here in gray are samples from French Polynesian outbreak. So there's this big outbreak in French Polynesia, isolated intense outbreak. You can see that it's in late 2013, early 2014. And then these are all the samples that we had at the time from the Americas. And what you can see then is that all the Zika viruses collected from all over the Americas, from northeastern Brazil, from southeast Brazil, from the Caribbean, from elsewhere in South America, in Central America, all of them have a common ancestor. They all appear to descend from a single introduction event from French Polynesia into the Americas, into northeastern Brazil in early 2014. And so basically every Zika virus that we've discovered of all the millions of infections in the Americas appear to derive from a single introduction event from French Polynesia to northeastern Brazil that happened in early 2014. Oh, yes?
What are the decimals that appear at the branches?
The decimals. Oh, yeah, good, thank you. So these are node support values yes, these are showing how confident we are in a few of these different colorings. So all of this requires kind of a statistical model behind it and so forth. And so basically as things diverge more and as we have fewer samples, we're less and less confident in our calls here of where we think things are. And so this 89 is saying that it's 89% confidence that this B node is in northeastern Brazil. The model says it could also be Haiti. So this is a Haitian sample that's kind of an outgroup here. We have now more evidence that it's not Haiti, but at the time it was kind of either northeastern Brazil, most likely, or possibly Haiti. And these, also thinking of confidence, these gray bars that you see kind of here are showing our confidence in the date that we've assigned to a node. So we think this is most likely to be around February 2014, but it could go back to late fall 2013 or as close as like, what is that, May 2014. So there's a bit of spread to these dates as well.
Do you have a cut point where you say this is so unlikely that we're not going to use it? I can't quite see how low all of the decimals go.
Oh, so everything gets a decimal. Everything gets an inference. We'll generally kind of, say this like 90-95% confidence is something you want to have. So in this case, at this moment, you couldn't say for sure that this node was, so let's rephrase what I just said. That we can't, at this moment, we couldn't be completely certain that it was a single introduction event from French Polynesia into northeastern Brazil. We have a 90% chance that it's a single introduction event from French Polynesia into northeastern Brazil, and a 10% chance that it's a single introduction event from French Polynesia into Haiti, and then a jump into northeastern Brazil immediately after that. Subsequent data where we've kind of filled out the tree more has confirmed that it's northeast Brazil. Other questions? Okay, thank you. So what's kind of very remarkable here, and one of the things that kind of the genomics really gives you, is that the first confirmed case of Zika that was kind of ever noticed in the Americas wasn't until June 2015. And so this occurred, and it was like the first case that we discovered. By the time we even noticed that Zika existed in the Americas, it had been there for a year and a half, and then it had spread all over the Americas. So there's all this cryptic transmission that we weren't noticing just because it wasn't on our radar. Zika wasn't something that was kind of known to be a threat. And so because it wasn't being noticed, none of these samples were being made, and so we wouldn't have had this story at all without the actual genomic sequencing. Okay, I'm just checking my time. I'm going to skip this next section. So we also looked at Zika in Miami. We have a bunch of other projects like this where we're looking at kind of MERS coronavirus epidemic in the Arabian Peninsula. And then now more recent work where we're actually doing a lot of sequencing ourselves. So Louise in the lab has been sequencing the mumps outbreak in Seattle and Washington State. There was actually a very sizable mumps outbreak last year that we've been kind of figuring out where it came from and how it's related. And then also more Zika work in the USVI in Colombia led by Ali. And so briefly, what we've been also working to do is try to make these analyses much more rapid and automated. And so what we need is something that rather than kind of waiting to publish a paper, and then that paper kind of takes a while to come out, and it has to go through this whole review process, which is like really important to have the absolute most correct science. For these issues of outbreaks, that always takes too long to kind of feed to public health. And so we're trying to make systems that can kind of get data out more quickly to people that can make relevant decisions. And so this is coming up with this, again, Nextstrain project. This is all in collaboration with Richard Nair. Then a number of folks from the lab are involved. James, Emma, Tom, John, Cullen, and Sydney have all been great here. Basically, we have two things. It's a bunch of bioinformatics to be able to do this stuff well and quickly, but also this website that we kind of keep a real-time display up to date of, which I want to give a very brief tour. So here we go. So here's the kind of basic page. We have different viruses that we're tracking. I'll just give you the example of how it looks with Zika virus that we're just going over. And so what you can see here is, oops, let's do it this way. So, so this is that phylogeny, basically, that we're looking at before I'm going to zoom in a little bit here, where we have these early samples in French Polynesia, as you can see here. So each dot that you're seeing is an individual person that was sick with the Zika virus. They had blood drawn, and then that virus was sequenced. And so there are these samples in French Polynesia, and then we have the whole spread in the Americas. But what we can do here is we can kind of wind back time, and we can look that if we're back in early 2013, it's only showing up in French Polynesia. But as then we go forward, we see it kind of moving and arriving into Brazil. And then from Brazil, we see it spreading into the rest of the Americas very, very rapidly. And so we can see this event of arrival into Brazil, and then spread from Brazil into Cuba, into Central America, et cetera. Then we get these secondary transmissions, for instance, from the Dominican Republic into Miami, which happened a couple of times, and so forth. And we can really kind of see the story of the outbreak unfolding. We can also look at other things like how different genotypes play out. So for instance, we have different mutations to the virus. This mutation is present in all of these viruses here, in this clade. This other mutation is present in these viruses up here in Central America. Oh, yes, go ahead.
You said that you saw the virus come to Brazil, but it was on the northeastern edge.
Yes.
Dots all in the middle.
Yes.
Is that not representative of where the sample is taken?
This is a good point. So what we've done here is I've just classified things as country. And so all in this case, all of the Brazilian samples got classified as just Brazil rather than Northeast Brazil, et cetera. And so then you have this arrival event as where does it go? Yes, right here, of arrival into confidence of 100% in this model to Brazil. We could break it up more in this analysis into like some things like, so like Colombia might just be labeled as Colombia and Brazil might be broken up because it's a large country. In what I'm showing here, I haven't done that. But that would obviously be something to do. Many of our other analyses, like I'll actually use the mumps example. So in case of mumps, in this case, we've broken it up at the state level. And so we can see in this example, this kind of event where we go from there was this outbreak in the Northeast. It actually moves into Arkansas, then bounces from Arkansas and causes the mumps outbreak in Seattle. And so we have, it's kind of a fair amount of flexibility, or maybe in one word, but the other word might be arbitrariness, in kind of what you choose for these kind of geographic bins. And so that's kind of a lot of choice on our behalf and the people working this field for kind of how to best break up geography. And there's not kind of a single clear way to do it. Okay, and we're slow on time. That's basically the website. We have a number of different kind of viruses here, and we're trying to basically keep all of this up to date, adding viruses as we can, so that if someone's working on Ebola or mumps or Zika or whatever, they have this kind of reference that they can always go to see what is currently circulating in the world. And is this going to be a bad idea? Whoa. Here we go. Okay. So generally, what's been really great is that this has been getting attention. We have even Bill noticing it. But what's been really fantastic is that this has been being used on the ground, as I mentioned. So people kind of working on Ebola, in this case, Sierra Leone, were able to kind of email us sequences every week, and we're able to get them on the website, and they're able to see where their viruses they were collecting fell. And with that, I'd like to thank people in the lab, a number of collaborators. Because I'm running low on time, maybe we should just skip to Jesse, and then we'll do more questions with both of us.
Hi, I'm Jesse. Just let me get this thing attached to me, and then I'll ask you if you can hear me. Can everyone hear me? Okay. So I'm going to talk about another aspect of looking at viral mutations. So I think Trevor gave a a really nice explanation of how you can use these mutations that are always occurring when viruses replicate to understand the relationships among viruses and build these phylogenetic trees. And he was describing how you could use this to look to see which virus is descended from which other virus and which virus moved from one place to another. So another aspect of looking at these viral mutations is you can also try to figure out what those mutations are actually doing to the virus. So you'll notice, for instance, in Trevor's talk, the mutations were just used from a historical perspective. He wasn't worried about what they were doing. He was just using them to reconstruct ancestry. So why do we want to understand the consequences of the viral mutations? So I'm going to start off by going way, way back. So this is this treatise written by this guy named Peter Panum. He wrote it in 1846, and it's called Observations Made During the Epidemic of Measles on the Faroe Islands. Does anyone even know where the Faroe Islands are? Where's that? Yeah, kind of. You certainly know better than I do when I started. They're up here, so they're kind of between Denmark and Iceland, maybe. And back at the time, they, I think, belonged to Denmark, and Peter Panum was a Danish sort of physician. And so you read this thing, and he kind of goes on in this very flowery language, which is not used in scientific papers very much nowadays. He talks about the winds, and he talks about throwing himself on the ground, and he talks about the northern lights, and so on. And if you persevere and you make it through about 5 or 6 pages of this, he eventually explains why he's actually at the Faroe Islands. And he says on the Faroe Islands there hadn't been any measles since 1781. And then the measles outbreak broke out in 1846, and that's why he'd gone there to investigate this. And the reason it had broken out after all this time is because the Faroe Islands were separated from the rest of Europe, and as Trevor was nicely describing, viruses move around, and nowadays Trevor was showing you how the swine flu moved across the whole world in a few months, but measles maybe was moving a lot slower back then because there wasn't so much travel to the Faroe Islands. And so he makes a lot of observations about this measles outbreak. But one of the things he notes is just how many people on the island got measles. So there were 7,782 inhabitants, and about 6,000 of them came down with measles. And then the thing that makes this a really interesting study is he also notes something about the 6,000 that did get measles and the 1,782 he didn't. So he notes of all of the aged people on the island who had the measles during this previous outbreak in 1781, not a single one of them, as far as he could find out, came down with measles a second time. And so I think all of you nowadays, sort of in the era of understanding immunity and knowing about vaccines, know why this is the case. He's describing immune memory. So it used to be if you got the measles once, you didn't get it again. Now if you get the measles vaccine, you're probably not going to get measles. And this is the basis of immune memory, and this is what protects us from so many viruses and why we no longer have lots of people dying from measles and smallpox and all the things that used to kill so many people. But something you might know is that we don't have good vaccines like the measles vaccine against all viruses. So for instance, you were probably all encouraged to go get your flu vaccine this year, and you were also encouraged to get it last year, and you're going to be encouraged to get it next year. And you're probably going to be encouraged to get it every year. So unlike measles, you can't just get this vaccine once and be protected for the rest of your life. And it also turns out, as some of you may know, it's definitely good to get this vaccine because it's better than nothing. But the flu vaccine is not like the measles vaccine in that if you get that vaccine, you're extremely unlikely to get the disease. There's still some chance, this happened to me 3 years ago, where if you get the flu vaccine, sometimes you still get flu because the vaccine isn't perfect. And so why do we not have a good vaccine against flu like we do against measles? So one thing you could look at if you were sort of coming from the perspective of an immunologist is you could say maybe there's something different about flu and we just sort of don't make as good of antibodies or a good of immune response to flu. And so we can actually look back in history and see if that's true. And the reason is there's sort of actually a historical example with flu that's a lot like Peter Panum's study of measles. So the history of flu, at least over the last 100 years, is like this. In 1918, there was a strain of flu called H1N1. It's also called the Spanish flu that came into humans, and it caused this big pandemic in 1918. And then that virus didn't go away. It kept evolving in humans for about the next 40 years. Then in 1957, there was a new strain of flu that jumped into humans. It caused what's called the Asian flu pandemic. And it evolved in humans for a little while, changing a little bit every year. Then in 1968, there was what was called the Hong Kong flu, and that virus still evolves in humans to this day. And so these viruses are reinfecting people every few years, and they're changing a little bit from year to year. But there was this really interesting thing that happened in sort of the history of H1N1 flu, where in 1977, a new flu strain called the Russian flu spread, sort of arose and started to spread around the world. And the virus that caused that actually was identical to a virus that had existed in humans in around 1954 or 1955. And it's actually thought that this virus was the result of some sort of inadvertent release. There's some speculation that it was due to some sort of vaccine trial, misguided vaccine trial going on among military recruits in China, where the virus that had actually been frozen in a freezer for these 24 years was reintroduced. So you now have something very similar to what Peter Panem was describing, where you have a virus that was here, and then all the people born after that date won't have seen that virus, and then the same virus comes back at some date in the future. So what happened in 1977? And in particular, just like Peter Panem noticed that there was this distinction between sort of older people who had measles before, they didn't get it in the 1846 outbreak, and the people who hadn't seen measles did get it, we can ask what happened with flu. So you would think that if our problem with flu is that we just don't make a good immune response to flu, then everybody would get this virus again in 1977, because no one would have a very good immune response to flu. If the problem is something else, in particular viral evolution, which I'll describe to you, then you might think that these people older than 24 will do just fine when the virus comes back, because they've seen that exact same virus before. But the younger people who haven't seen that virus might have problems. So if we look what happened in 1977, here's an article from the British Medical Journal. It's describing influenza in a boarding school. So they use much more succinct language than Peter Panem. They get right to the point. The Easter term had begun in January, and they'd had a boy from Hong Kong, so someone just like Trevor was discussing, carrying a virus and traveling from one place to another, that had returned to the boarding school. He got sick in the middle of January, and by late January, they had more kids who had the flu, and overall, 512, or 67% of all of the boys in the school got sick and spent days away from class. But of all the adults that worked in that school, only one of them, a house matron, came down with that strain of flu. So this is really showing you that the immunity against flu that at least you get from these natural infections is just at least almost as good as the immunity against measles. This natural infection was protecting these people 24 years later. But the key point, it was only protecting them against that exact viral strain. So the reason these older people were protected is because they'd seen that exact strain or something very close to it 24 years before. And so the reason that we're at risk from flu virus every year is not really due to some deficiency in our immune systems. We can mount good immune responses against flu. The reason that we're susceptible to flu every year, every few years, is due to this viral evolution. So it's due to these changes in the virus, like Trevor was talking about at a genetic level. And we actually understand how this works at a molecular level as well. So when we look at these viral mutations, are actually happening in genes in the viruses, which make proteins. So if you were to look at a flu virus, sort of the bottom of the virus, or the part of the virus that has all the genetic material would be down here, and this thing is sort of sticking out of the virus. And there's a whole bunch of these proteins, which are called hemagglutinin, several hundred of them, that are sticking out of the virus. And so if a person gets infected with flu, they make these really good, really potent, really powerful antibodies, like this antibody shown here in red, that bind to this hemagglutinin protein. And these antibodies do a really good job of protecting you against being infected with that strain of virus again. And so probably the reason that all the adults in that boarding school, and for some reason this one house matron, were protected in 1977 is because they still had these antibodies around from 24 years previously. But unfortunately, something that's a property of a flu, even though it's not a property of all viruses, it's not so much a property of measles, is that if a virus is exposed to an antibody or subjected to selection from an antibody like this for a little while, the virus will quickly get mutations. So here I've shown examples of some of the viral mutations you can get here shown in green, right where the antibody binds, and then the antibody doesn't bind anymore, and the virus can get away from that antibody. And then when we look at flu over long periods of time, so for instance, if we look at this H3N2 flu, which has been in humans from 1968, and we say, where are all these mutations that have happened from 1968 up until now, you can see they're all over the surface of this protein, especially up here on the top where the antibodies tend to bind. So this is the reason that we don't have this really long-lasting immunity to flu, because everywhere that the antibodies are looking at the virus is changing. And so when we think about this virus, there's sort of two major selective forces that are driving the evolution. I've talked about the antibodies, but that's not the only thing going on. The virus has to keep being able to grow, so this hemagglutinin protein isn't just on the virus to enable antibodies to bind to the virus. It does something important for the virus. And it has to maintain that function so the virus can keep growing. And then the other thing that the virus wants to do is accumulate mutations that evade antibodies. So one of the things that we're really interested in doing is mapping how these mutations to the virus affect these properties, such as how well does the virus grow, or how is it recognized by the immune system. Because we'd like to then take the types of genetic analyses that Trevor was describing and lay on top of that the ability to not just use the mutations to reconstruct how the viruses are related to each other, but maybe understand which viruses have mutations that might affect things that we care about, like such as does our immunity recognize that virus well. And so we do that by trying to do experiments in the lab. So as Trevor was describing, the viruses out there in nature are getting these mutations, but they also have these very complicated evolutionary relationships. So anytime you see a mutation, for instance, in the case of Zika virus, you don't know if Zika virus has that mutation because that mutation is somehow making the Zika virus better, or if it just happened to be present in that virus that got transmitted over to Brazil, because there's a lot of random things going on during evolution. So what we want to do is do experiments that sort of get rid of those random factors. So again, going back to the idea of the protein flu hemagglutinin as an example, this protein is just encoded by some gene which encodes 566 amino acids. And so in the lab, using modern techniques, we can just make all of the different mutants of this gene. So there's 566 times 19 mutations, which is about 10,000, or exactly 10,754, if I didn't mess up my math. And that sort of sounds like a lot, and it's definitely a lot if you're trying to make each one at a time to look at it. But with modern techniques, one of the things that's happened is it's gotten sort of more high throughput in biology, and there are ways that you can sometimes look at lots of different things all at once. And so we're able to generate all of these at once, and then what we do is we just use them to generate a pool of viruses in the lab that now have all of these 10,754 mutations in them. And then we can just ask, how do those mutations affect some property of the virus that we can control in the lab? So for instance, we can take all these viruses and we can put them in a dish and ask which ones can grow well. And so the key difference in doing this versus looking at the natural evolution like Trevor was doing is this doesn't tell us anything about how the virus is evolving in nature, because the virus in nature may get different mutations than the ones we put in here, or may not get all the mutations we put in here. But unlike in nature, there's not some complicated evolutionary history where one of these viruses got lucky and was on the airplane or the cruise ship or whatever that went to Brazil. They're all there. So the outcome is really dependent on the features of these viruses and not the chance so much of evolution. So what we can then do, and this again is using modern techniques, is we can now use sequencing, so the same type of sequencing that Trevor was talking about, to sort of sequence all of the mutations that we started with, and then all of the mutations that are present in these viruses after we do some sort of selection. And then you can just look and see what are the effects of the mutation. So if we just look at an average level, you probably all know that when you make a mutation to a gene, it can sort of have three different properties. It can be what's called synonymous. So it changes the nucleic acid sequence. And in the case of our genomes, that's DNA. In the case of flu, it's RNA. So it changes the RNA sequence, but it doesn't change the protein that RNA sequence encodes. So that's called the synonymous mutation, and usually they don't have so much of an effect. It can somehow change the amino acids or the letters in the protein. That's called the non-synonymous mutation. And often, but not always, those mutations will have some effect on how well the protein can work. Or it can put a stop codon in the protein, and that's pretty much always bad for the protein, because then you're just not making the whole protein. And so when we look and we see what mutations are present in the viruses that actually grow, we see sort of what you expect. These stop codons, which are always bad, go away. The synonymous mutations stay about the same. And some, but not all of the non-synonymous mutations change in frequency, because sometimes, but not always, these mutations will have an effect. And then the nice thing is we can perform this sort of analysis that I've drawn out here at an average level. We can do it for every single mutation to the viral genome. So I am going to skip a few slides here and just talk about using that method to describe the thing that I think is most interesting, which is how do these mutations affect how well our immune system can recognize the virus. So we can now use this to completely map how mutations would affect the recognition of a virus by any given antibody. And so we do that just like I was describing. We take and we make this pool of viruses in the lab, just like I was describing on the previous slide, and then we do two things with them. We either treat them with some sort of antibody, and this is sort of supposed to mimic how immunity would select them, or we maybe don't treat them with an antibody. And then we just look and we see which viral mutants are here versus here. And here you would expect any virus that can sort of replicate will be present, and here only the viruses that can replicate and get away from the antibody will be present. And then so we just do sequencing to read that out, and then I'll kind of represent those data like this. So, DNA encodes amino acids, which can be represented as letters, and when I draw a big letter, it means at that particular position in the protein, that letter, so in this case D is aspartic acid, helps the virus get away from the antibody. And so we wanted to sort of try this method out. So the first thing we did is we worked with a collaborator, not Trevor, another collaborator named Scott Hensley, who had antibodies that they, I think in conjunction with other collaborators, had actually isolated from people who'd received the flu vaccine in 2009, 2010. And the reason we did that is the strain of virus that we were using in the experiments I described to you was a virus from 2009. And then we just looked and we saw which mutations were selected by that antibody. And so down here are these little letters you can see. So basically across the whole length of that protein, which is 565 amino acids, the antibody pretty much only selects mutations at a few sites. So those are shown here. There's just a couple of sites where most of the selection is targeted. And if we go and we look at the structure of that protein, we can see that those sites mostly fall up here on the top of the molecule. So that sort of tells us that's where the antibody's binding, and that's why mutations there help the virus get away. And we can do this with other antibodies that were isolated from this same individual. And you can see that the different other antibodies are selecting mutations at slightly different positions, both in the sequence and on the structure. And here's another one. And this is something that's known. It's known that in our bodies we have lots of different antibodies, and the different antibodies will bind to slightly different places. And then a really cool thing that we can do is we can then go to that Nextstrain website that Trevor was talking about and go look at the evolution for flu, which I don't think he specifically showed, but we can, in his presentation, but we can go and we can look at specific sites in flu and see what happened. So this is, for instance, this antibody strongly selects mutations at site 160. In fact, the mutation that's most strongly selected by that antibody is this mutation from lysine to threonine. And we can, in fact, see that during the evolution of the virus, including around 2009, when this antibody was isolated, the virus had a lysine there, but a few years later, a threonine appeared. And actually, this is showing that by 2018, almost all of the flu viruses circulating in humans now have that threonine. And we can sort of do this for these other antibodies as well. So this antibody selects mutations at site 159, and sure enough, there are mutations at site 159 that appear during the evolution of the virus, and the same thing is true here. Now, importantly, this is sort of very anecdotal and slightly cherry-picked examples, but it's showing that these antibodies are selecting many of the same mutations that occur during the natural evolution of the virus. So this is sort of very exciting because we can try to relate these mutations that are occurring in nature back to what they might be doing. Importantly, I was just describing 3 antibodies that were isolated from one person, and any given person has a lot more than 3 different antibodies in their body. I don't know what the estimate is, but it's going to be like many, many, many orders of magnitude more than 3. And then there are also a lot of people in the world. And so one of the things that's very exciting now is to imagine, can we take the actual serum from people? So if you just take blood, there's a whole mix of antibodies in there, and do these same sort of selections with sort of mixtures of antibodies, which will all be targeting different parts of the virus, and see what happens. And so we're just starting to do these experiments now. Actually, Juhei, who I think decided to come suffer through this, even though this talk is mostly just slides I stole from her, is the main one doing this research. And I think in addition to sort of seeing what sites the antibodies from people select, this will allow us to address sort of a really interesting and long-standing question, which is how does the virus actually get away from antibodies when real human flu is evolving? So I described how you take one antibody, the virus gets a mutation, it gets away from that antibody. But I also pointed out that real people have more than one antibody. And it's not so obvious how this is going to work when you have a lot of different antibodies. So the virus might get some mutation, and maybe the blue antibody can't bind anymore, but what about all of these other antibodies? And so we don't really yet know the answer to this, but we're certainly not the first person to think about this problem. So for instance, there's this paper from several decades ago now by John Udell Rob Webster and Walter Gerhard, and this paper was actually published. Walter Gerhard was one of the people who was among those to sort of first develop the ability to isolate antibodies from humans, or I think in this case actually mice, and they used it to look at what were the effects of these antibodies on people. And they found that the antibodies could target at least 3 different parts of the flu hemagglutinin. We now think they could target even more than 3 parts. And so they were aware of the same sort of mystery I was talking about, and they were wondering, how does this antigenic evolution actually happen in humans? And they wrote a whole bunch about it here, which we won't all read, but one of the things they suggested is perhaps there are certain individuals, so certain people in the population, who possess an antibody repertoire that is really not binding to all the different parts of the molecule, but is really focused on just 1 or 2 epitopes. And then you could imagine maybe the viruses in those people could sort of more easily get mutations to get away from that very restricted part. And then maybe if the virus transmitted to someone else who had antibodies targeting a different part, it could get away there. So that was their speculation, and I don't think we really know the answer yet, but it seems like this might turn out to be pretty good speculation. So just to sort of conclude, the key points from my part of the talk is that we can look at these mutations and we see what they're actually doing. And we know in the case of flu, what they're allowing the virus to do is escape antibodies while still maintaining its ability to function. We're now starting to develop techniques where we can really map how these mutations are affecting things like the function and the immune recognition of the virus. And we think this is really exciting because sort of integrating these functional and antigenic measurements with the types of genetic analyses Trevor was describing could allow us to better understand how and sort of why these viruses are evolving in nature. And then, like Trevor, I'd like to end with some thanks, particularly to Mike and Juhei, who are the 2 grad students in the lab who did this work, Trevor and a grad student in his lab who've sort of helped with analyzing natural evolution, and then Scott Hensley, who is a collaborator of ours at University of Pennsylvania, who is a person, for instance, who gave us the antibodies that we were using in that study. And then my funding. Thanks. So I think we can sort of both take questions now, if anybody wants to stick around for the last 5 minutes, although you should feel free to leave as well.
Oh, yeah, no, sorry, down here.
So, great talks. So I have a question about the Nextstrain, and maybe it goes with what JC says, viral evolution. How can you use basically what you know to actually predict? Because I don't think you talked about predicting the next strain.
Trevor, do you want to use this thing?
So that kind of falls into 2 categories. There's a whole field that's trying to do the prediction of if Ebola is in these different regions, how can it kind of go forward from there. We're not doing as much with that. That's quite hard, and it requires knowing how very detailed levels about how humans are moving about the world, et cetera. We're working much more on the predicting which strains of flu will take off, and so there we're using kind of these different predictors of which sites have mutated and so forth. We're actually doing forecasting there, and then we're working with Chessie to kind of pull in those experimental results to improve that forecasting and try to actually make it next rather than just now.
Yeah, catch.
Mike. To Jesse, how do you model indel mutations when you're generating your mutants?
Oh, indels? Yeah, so the question is, when a gene evolves, it can get point mutations that I was talking about, and it can also get insertions or deletions where it adds or takes away DNA, or in the case of flu, RNA. So actually, for the particular case of flu, we don't. And the reason is that it just happens that the way flu evolves is it has very few insertions or deletions. That's just kind of a feature of the evolution of the genome. So most of our experiments so far have been focused on flu, and flu, if you look all the way back from 1968 till now, the hemagglutinin gene has not gotten any insertions or deletions in humans. But if you were to study certain other things, like for instance, HIV is another virus, and it gets a lot of point mutations, but it also gets a lot of insertions and deletions. And certainly when you look at the evolution of human genomes, insertions and deletions are pretty common, or duplications are pretty common as well. So we're not really worrying about that right now because we're mostly focused on flu, but I think if you were to want to study the evolution of sort of entities that had a lot of insertions and deletions, you'd want to put those in your library as well.
The question is also for Jesse. Have you found instances where there's RNA interference going on with maybe a non-synonymous mutation?
Yeah, you mean so for instance
I mean,
where
it's synonymous.
you mean where a synonymous mutation has an effect? Yeah, no, that's a really good question. So I mean, I gave this very sort of simplified view here where it was like the synonymous mutations aren't doing so much, but in reality, the flu genome itself, which is this piece of RNA, has structure, and sometimes the synonymous mutations could influence the structure of that RNA. And I know in certain cases, known those can be really bad for the virus because certain aspects of the structure and the packaging of the RNA are really important. We haven't looked at that so much. I think it's much more common for the mutations to have effects by amino acid mutations, and we haven't tried to analyze a lot for synonymous mutations, but it is known from other work that in some cases synonymous mutations, particularly at the ends of the genes, can have big effects. And the reason is because the virus uses the exact nucleotide, not protein sequence at the end of the gene, to package the genome into the virion. So there are certain positions there that if you mutate, the virus just won't be able to get the gene into the virion, even if the protein that the gene encodes is just fine. But we haven't focused on that so much at this point.
I'm not going to throw this at you.
Well, no, do the stop. It was just that there was no more.
So my question is less about the science itself and more about how do you all decide to collaborate and the point at which like you say, oh, your work intersects with my work and will inform, and together we will actually make something better. So, I mean, maybe you'll talk about like a specific example of what you're working on together that brings your work together.
Do you want to go?
Go ahead, Jess.
Okay, well, I think a good example, I think it's very useful to collaborate with people when they have clearly complementary expertise. So in the work that I talked about there, for instance, we're looking at the effects of these mutations and, then we want to see did those mutations actually happen in nature. And I mean, sure, we could go try to figure that out ourselves, but Trevor or John Huddleston in Trevor's lab already has a lot of expertise looking at that, so it works really well to just, kind of go work with them to find that out. Or in the case where we wanted those antibodies from humans, I mean, I suppose could have tried to set up some protocol to clone them ourselves, but it would have taken us a long time, and Scott Hensley just has those antibodies. So I mean, I think you want to collaborate when people have complementary expertise. In order to collaborate, you also sort of have to know people and trust people, right? I mean, you don't want to go to all this effort if someone like isn't actually going to follow through, or you don't think they're going to do a good job, or I mean, you have to have a lot of trust, right? Because you're putting a lot of effort into something, and then you're expecting someone else to also do the same thing. So I think the other big aspect of collaboration is just knowing the people that you're collaborating with, and sort of and sort of talking about how projects will work. And so, for instance, one of the reasons that we collaborate both with Trevor and even Scott Hensley, who's at Penn out in Philadelphia, is like we all kind of got to know each other like 2 summers ago. Me, Trevor, and Scott all did like a retreat, where everyone in the lab came and kind of, met each other. And I mean, for instance, Juhei, there she got to know pretty well both this guy Seth, who was a grad student in Scott's lab, who actually had those antibodies, and then so it was very easy for her to email him, and he sent out the antibodies. And then she also got to know John, who's a grad student in Trevor's lab, so it was very easy for her to like go talk to John and like ask him what he thought about stuff. So I think it's a combination of identifying complementary expertise, but also I think the Hutch is a good place for collaboration because it's fairly small and you'll sort of know people, and other people are pretty nice, so you kind of trust them and feel excited about working with them.
How did that retreat happen? Like, what initiated the retreat itself that sort of nucleated this wider?
Decided like we should do this?
Yeah, we were at there was some big flu meeting in Chicago that a bunch of people from my lab went, and a bunch of people from Scott's lab were there, grad students. And it was one of these meetings that turns out to be like more tailored towards sort of like the famous people who are going out to fancy dinners. But as it was, I think the grad students in my lab and the grad students in Scott's lab were all kind of hanging out. And then, Scott fortunately has a good sense to realize that Seattle's a much nicer place than Philadelphia. So we would not have wanted to spend a summer flying to Philadelphia, but they were very excited to spend a summer flying to Seattle. And then we were able to have a retreat there.
I guess sort of related to both talks, I guess I'm curious, maybe Trevor, with the mumps outbreak where we do have a vaccine, is it that the mumps is escaping it, or is it this now related to people not vaccinating anymore?
So that was kind of an open question, and we've been having a bunch of mumps outbreaks happening all over, like not huge, of the span of 2,000 cases in Washington, 5,000 cases in Arkansas, about that many cases in Massachusetts. And what seems to be happening is it's actually waning immunity. So it's that we have a 2-dose kind of mumps vaccine, and that is the least effective of the MMR, of mumps, measles, mumps, rubella vaccine. So naturally, it's only like 80% effective, something like this, 85. And so that should be enough to bring up herd immunity, but basically what we see in these outbreaks is it's not the kids, they're all fine. It's people once they're about 18 or so that their antibodies have waned enough that they're able to actually get infected. And then so we're seeing these mumps outbreaks are occurring in kind of situations where people are having a bit more contacts with each other than you'd expect in other cases. So it's like college campuses, the hockey team is one example. In the Seattle case, it was actually kind of this, what we're using for kind of on the public side, a close-knit ethnic community of kind of a group that is like very, has kind of a lot of familial ties.